Thank you, Dr. Sawyers. Thank you very much for inviting us to present here. On behalf of myself and my colleagues, I will be discussing our phase one first-in-human trial of DMUC 5754A in platinum-resistant ovarian cancer. Ovarian cancer is the leading cause of death from gynecologic malignancies in women in the United States, with approximately 22,000 cases and 16,000 deaths occurring annually. Although initially chemotherapy and surgery for this disease can be quite effective, most cancers will recur and become increasingly resistant to further chemotherapy. These cancers, often termed platinum-resistant, are a particular challenge to treat, and current therapies for them are limited in their effectiveness and typically short-lived. DMUC 5754A is an antibody-drug conjugate, which is basically a DMUC 5754A is an antibody-drug conjugate. It's a type of drug where you have a monoclonal antibody that identifies a particular target linked to a potent toxin by a cleavable linker. The structure of DMUC 5754A is shown in this slide. The antibody portion of DMUC 5754A here identifies the protein MUC16, which is highly expressed in certain cancers, including approximately 80% of ovarian cancer. Of note, when cleaved, the extracellular portion of MUC16 is released into the serum as CA125, which is a well-established biomarker for ovarian cancer. The anti-MUC16 antibody is linked to the toxin monomethyl auristatin E, also called MMAE, which disrupts microtubules and causes cell death. As illustrated in this slide, this is the mechanism of action of DMUC 5754A. It binds to MUC16 on the surface of the cell, and upon binding is rapidly internalized. MMAE is then released into the cell where it causes microtubule disruption and cell death. This study was a first-in-human phase 1 trial of DMUC 5754A in platinum-resistant ovarian cancer, as I've mentioned. 44 total patients were enrolled to the study over 6 dosing levels. Overall, women participating in this study were heavily pretreated and had on average 7 prior chemotherapies for their cancer. As part of the study, we looked at expression of MUC16 in an archival sample of each patient's cancer and asked if this could be a possible biomarker of response to DMUC 5754A. We found that given on a once every 3-week basis, DMUC 5754A had an acceptable safety profile, with most adverse events attributed to the drug being grade 1 or grade 2 in green and blue here. Observed adverse events at the maximum tolerated dose of 2.4 milligrams per kilogram included fatigue, nausea, and peripheral neuropathy. In the 29 patients treated at the maximum tolerated dose, the number of severe toxicities attributable to the drug was pretty small, with the most frequent being grade 3 fatigue. Here. This slide illustrates the activity we saw with DMUC 5754A in patients treated at the 2.4 milligram dosing level. This is a waterfall plot, so bars that rise above the 0 axis represent tumor growth, and those that fall below the axis represent tumor shrinkage. As you can see in this plot, a significant number of patients did have shrinkage of some degree in their disease, with 4 patients having a partial response, meaning a shrinkage in their tumor of at least 30%, and 1 patient having a complete response, meaning they had complete resolution of all visible tumor. One of the things that we also did, as I mentioned, was we tested IHC expression of MUC16. And you can see that those with very high expression, 3+, are here in this orangish color, and 2+ in the teal. All 5 responders did have high expression of MUC16 in their tumors. This slide summarizes the overall results of our study. We found that DMUC 5754A had an acceptable safety profile, with most adverse events occurring to be grade 1 or grade 2. Fatigue was the most frequently observed grade 3 or 4 adverse event. Peripheral neuropathy, which is a toxicity that has been previously associated with this antibody-drug conjugate platform, was manageable and reversible in most patients with dose delays and reductions. The pharmacokinetic data, which I haven't presented here, showed that The PK was not affected at our 2.4 milligram kilogram dose by the serum CA-125 levels. We observed anti-tumor activity in platinum-resistant ovarian cancer with 1 CR and 4 PRs, and we had additional patients who experienced minor responses with prolonged stable disease. As a preliminary biomarker, we saw responses only in patients who had high expression of MUC16. This is the first presentation of a drug in this class in ovarian cancer, and these results are encouraging in terms of activity. If the activity of DMUC5754A is confirmed in additional trials, this would represent a potential novel therapy for a type of ovarian cancer that is quite difficult to treat. Thank you.